All right, well, hello everyone. My name is Iman Khwaja Reddick. We are pleased that you could all join us for this week's lecture in Volume 3 of the Know Neuroanatomy Didactic Series that brings you lectures from experts in the field covering different topics each week. This series was created by trainees and early career neuropsychologists, including standing members of the KnowNeuropsychology Board, as well as the KnowNeuropsychology Committee. We would like to thank our 2023 sponsors for their generous financial support of this series. Before we start our lecture, one of the main goals of KnowNeuropsychology is to provide free, high-quality didactic content to our audience. Every KnowNeuropsychology and KnowNeuroanatomy lecture is available on our YouTube channel. Be sure to check it out, subscribe, and like our lectures to get first access to the new content. New to KnowNeuropsychology is our collaboration with APPCN to bring you learning and discussion questions that are provided with specific lectures content. You can access these on our website and through APPCN. Here are the disclaimers for the series. This training is not meant to replace formal education in neuropsychology, and the views of the speakers are their own. Questions can be submitted via the Q&A box on the lower left of your screen, and a recording of today's lecture will be provided on our website and YouTube channel later this week. Now it is my pleasure to introduce Dr. Eva Geringer for today's lecture titled Subcortical Synthesis. Dr. Geringer teaches clinical neuroanatomy at the University of Oklahoma School of Community Medicine in Tulsa, Oklahoma. Over her career, she has taught a wide range of students in various capacities, both MD and DO students, physician assistant students, doctoral nurse practitioner students, postdoctoral fellows, residents, graduate students, undergraduates, high school students, grade schoolers, and even preschoolers. After doing research in electrophysiology lab to obtain her PhD in biomedical sciences, Dr. Geringer chose to work as a part-time instructor in Oklahoma State University Center for Health Sciences Neuroanatomy course. She has a passion for education, particularly in the medical field, and is always delighted to create ever more focused and efficient lecture materials and study tools to facilitate high-volume learning. She particularly enjoys working one-on-one with students in both the classroom and lab, and gleefully takes any and every opportunity to show brains from her specimen collection to anyone who wants to see them. We are so excited to have you and cannot wait to learn from you today. So I'll go ahead and turn it over to you, Dr. Geringer.
All right, thank you very much. I'm very excited to be here. Let me go ahead and share my screen. Here is, and this is a this is a massive topic. I've watched the previous topics in the series and have attempted to integrate what the other presenters shared with you all. And then the way I've divided this up is the first part of this talk is anatomy, like really focused on anatomy and cross-sectional anatomy and correlating that with MRI imaging, because I always say if you are seeing actual brains in your practice, neuroanatomy is probably not your main focus at that point. It's usually imaging is where you're going to see the brain. And then, if you have questions during this initial part, go ahead and send them in, and then we'll have a short Q&A about anything you have particular questions about, which is the anatomy. And then the second part of the talk is about the specific neural circuitry and with clinical correlations put together with it. And I have a ton of content that is just how I do things. My husband always says overdoing it is the way you do it. So with that being known, we're going to get through what we get through. I will make this talk available so that the content is available if we don't get to things and people want to take a look at it. All right, so with that said, let us move along on our subcortical synthesis. So major subsystems that are influencing cerebral cortex and the periphery that were talked about in this lecture series: hypothalamus, which short version, that structure is involved in homeostasis via outflow generally to the pituitary gland, to the brainstem. And then I know Dr. Moreno said limbic system is, I guess, not exactly supposed to be used anymore, but I don't have another term for that. So I'm going to go ahead and call it the limbic system. Then the amygdala is a set of nuclei that is under this structure called the uncus that is incredibly important, essentially in threat assessment, placing an emotional valence or value on our experiences that we have. And then it's also, you can't have fear conditioning without an amygdala. How does that happen? Well, it is via outflow to brainstem nuclei, outflow to the basal ganglia, which is coming up, and then the basal forebrain and the limbic system as well. Then the hippocampal formation is a structure that underlies this gyrus called the parahippocampal gyrus, and its major job is memory consolidation. It's also involved in spatial orientation. How does that do that? Well, that outflows to the hypothalamus, so the mammillary bodies specifically, and then also to the basal forebrain. Basal ganglia, these are a collection of nuclei at the base of the forebrain, and their job, the way I say it, is they gate cortical activity, and the regions of the brain that they're making sure work at a proper level are found in the frontal lobe, so motor cortex, also prefrontal cortex, and then also in the limbic lobes to make sure we have appropriate levels of motor, cognitive, and emotional function. Some of the cortical regions that I want to just make sure we know what we're talking about before we get into the functions, since these systems, not only are they regulating what's going on in the periphery, but they're regulating cortex. So what cortex are they regulating? Prefrontal cortex, kind of a big deal in terms of human behavior. So dorsolateral prefrontal cortex, this is for executive function, and I think of it more as like outward executive function. So order and initiative. Dorsolateral prefrontal cortex is also really important in kind of that ability to just hold ideas in your head to work with them. So this thing called working memory. So I think of dorsolateral prefrontal cortex as my do it cortex. So let's make a plan and let's follow through. Kind of what would Brian Boitano do? He would use his dorsolateral prefrontal cortex. Orbitomedial prefrontal cortex, I think of this more as inward executive function. So the things that basically make us good social creatures instead of just being very productive. So restraint, social intuition, and evaluation. So orbitomedial, I think of as like the oh, can I stop myself cortex. So this is just a chart showing kind of general functions of prefrontal cortex. Restraint, order, initiative is usually how that is explained. And the oddity of prefrontal cortex is if you mess it up, you can kind of go two different directions. So if we've got a problem with dorsolateral prefrontal cortex, that can manifest as abulia, so the word ebullient means, you're kind of all out there and very externally focused. Well, abulia, we have no more buell. So we just have no motivation. The individual may look like they're paralyzed, but they're really not. They're just sitting there quiet because they just don't have that external engagement. Alternately, disorder with the orbitomedial prefrontal cortex can cause disinhibited behavior that I think of it as, oh, no, I can't stop myself. So we have symptoms like confabulation, where you just don't really tell the truth, even though you should. You've got emotions up and down, kind of all over the place. Then anterior cingulate cortex is another region of cortex that comes up a lot when you're talking about these subsystems. And this specific region of prefrontal cortex is for selective attention and motivation, and disorder of this structure can cause what's called mutism, where akinetic mutism, where they just sit there and do nothing. So similar to abulia, but a little bit more excessive, talking, And then last region that does show up is this cortex called the insula, which is really fascinating cortex, but this essentially puts together a whole bunch of kind of our internal interoception. And then it's also important in empathy as well. Okay, so that's just kind of a setup. Now let's talk anatomy. So here is, when I'm teaching anatomy, I tell my students, first thing you want to do is orient. So what we are looking at here are axial or horizontal section through cerebral cortex. And so I always get mad at my order of slides, but usually I think a great place to start is the ventricles. They're pretty good for orienting. And let's see. Okay, so here's my ventricle. Here's my space. So anteriorly, this whole region is lateral ventricle. This will be the frontal horn of the lateral ventricle. And yeah, this is animating these things is annoying. So frontal horn of lateral ventricle. And then anatomically, if you will look on the lateral wall of the lateral ventricle, you're going to find a structure called the caudate nucleus, which is part of the basal ganglia. So here we see this opening of the lateral ventricle, and we see the caudate nucleus. The lateral ventricle is a C-shaped structure. The caudate nucleus is a C-shaped structure as well. So here we're seeing the head of the caudate, and none of this actually existed before yesterday. So this is, you get to see my rapid editing when I see things wrong. So here's the body of the caudate. Then medially, we're going to find this structure called the thalamus, which I call the balls in the middle of the brain because they're roundish and they are right in the middle of the brain. And then lateral to the caudate nucleus, we're going to see some white matter. So at this point, before we're seeing any of the other nuclei of the basal ganglia, this is actually called the corona radiata, but then that is, and when we descend down, we're going to see those axons are going to coalesce into a structure called the internal capsule. Okay, so there's where I finally animated my lateral ventricle. If we look over here on this corresponding section, we should be able to see similar things. I guess I also went ahead medial to, so here's the caudate nucleus. On the medial surface of the caudate nucleus, there is a tract called the stria terminalis. And notice my colors, I kind of obsessive compulsively make basal ganglia are orange for whatever reason. The thalamus gets rainbow colors because it is relaying all sorts of things. And then amygdala ends up being this dark purplish. So the stria terminalis is actually outflow from the amygdala. And then anteriorly, you'll actually see a hole in it. There's literally a vein in there. It's called the thalamostriate vein, and it is draining the thalamus and the striatum, which refers to caudate and putamen. All right, so let's see if we can find these things over here. So on an axial section, I always think the frontal horn of the lateral ventricle it has kind of this very characteristic squashy oval shape. And then towards the back, this would be the atrium or the big open part of the lateral ventricle. This hits the thalamostriate vein really, really nicely. Actually, the thalamostriate vein is going to be in the stria terminalis, and then it takes this little bend around and forms the internal cerebral veins, which end up draining into the great veins of Galen and drain into the straight sinus. So that's just when we do a dissection, you end up pulling all of that stuff off. Lateral wall of the lateral ventricle, here we see a lovely head of caudate, and then it kind of narrows down into the body of the caudate. We can see the lump of the thalamus medially. And then this would be the white matter of the corona radiata, and we can see cortex, cerebral cortex all around here. Other note, this is particularly when you're trying to learn anatomy on MRI, I think a really good way to do it is with T2 weighted MRIs because the cerebral spinal fluid shows up bright, bright white. And I just think the contrast is better, and blood vessels just kind of pop out because they're dark against this white CSF a lot of times. So that's just, I guess, my advice to you. All right, so we're going to just head down. Basically, we're going to go from where we started and go down to the midbrain and identify structures. And hopefully this stuff will start to look a little bit more familiar. So again, first step, you always want to orient. We are looking at a horizontal section. We have horizontal wet specimen and then a horizontal T2 weighted MRI. And again, I like to start with kind of the brain holes, and you can see here the characteristic squashy oval shape of the frontal horn of the lateral ventricle. Back here, we have the atrium of the lateral ventricle. And you see here, this is actually MRI. A former student gave me permission to use this MRI. So from a young 20-something-year-old male. And you can see these specimens are from anatomy donors. And so their ventricles tend to be a little bit bigger because they're just older. We've got some neurodegeneration, and that means bigger spaces. So here we see frontal horn and atrium of lateral ventricle, and then lateral wall of lateral ventricle, we will see the caudate nucleus. So here we see the head of the caudate, and actually we see the tail of the caudate down here associated with the atrium of the lateral ventricle. And then also part of the nuclei of the basal ganglia proper is a nucleus called the putamen. And here we see putamen, and over here you can see, oh, it's the same color as the caudate. Embryologically, they're derived from the same structures, and so they actually look very similar. Then we do have some white matter in between the caudate nucleus and the putamen. And so at this point, instead of corona radiata, now that we're seeing putamen, we can identify 3 different parts of this internal capsule structure. So the anterior limb of the internal capsule is between the head of the caudate and the putamen. The genu of the internal capsule is actually at the level of what's called the interventricular foramen between the lateral and the third ventricles. And then the posterior limb of the internal capsule is lateral to this structure right here, which, oh, that's thalamus, that falls in the middle of the brain. All right, so here's putamen, and then also associated another C-shaped structure that is basically, we're always going to find it medial to the caudate nucleus, is the stria terminalis, which is outflow from the amygdala. And then this kind of anterior regions are where we're going to have the little hole in it that is the thalamostriate vein. So over here, put these same structures in on the MRI as well. Oh, and that's actually, I get excited when I see internal cerebral veins. Don't ask me why. All right, so then our next ventricle here is the third ventricle, and you can see on this individual, it's definitely a slit-like ventricle when the brain is kind of all still there and fully accounted for. Lateral to the third ventricle, here is our thalamus, and here is thalamus right here. And then other structures we're going to identify are associated with the hippocampal formation and memory consolidation. So that thing called the circuit of Pepez that Dr. Jennett talked about. And so here we're seeing portions of the fornix, which is also a C-shaped structure. And so we're seeing kind of posterior and anterior parts of this as it's curving around from the hippocampal formation. And I'll have some pictures of that as well. Also important in memory formation, so you see there's some kind of nuclei. This structure is the septum pellucidum between the 2 lateral ventricles. Anteriorly, there are some nuclei in the septum pellucidum that are cleverly called the septal nuclei. And these are cholinergic nuclei associated with the basal forebrain, and actually are very important in memory consolidation as well. So do we see these same things over here? Well, here is some back of the fornix. There is some fornix at the base of the septum pellucidum, and we can see, yeah, there are some nuclei in the septum pellucidum as well. Insula, it's basically going to be on all these slices. I won't identify it on everything, but essentially, once you start to move your way out laterally, you're going to hit, if we're far enough back, we'll hit thalamus, posterior limb of internal capsule, basal ganglia, and then the next gray matter we get to of note is the cortex of the insula, deep in the lateral fissure. All right, so now we're moving on down. So we're seeing a few more fancy things with the basal ganglia here. Always, though, I think it's good to start with the ventricles or the hole. So here we see the characteristic squashy oval shape of the frontal horn of the lateral ventricle, and we've got a nice big atrium of the lateral ventricle back here. If we, I guess, okay, so at this level, we are also seeing the interventricular foramen between the lateral ventricle and the third ventricle. So if we look over MRI-wise, what are we seeing? Well, here's frontal horn, but there's not much of it left. And so if you look at a little diagram here, of the frontal horn of the lateral ventricle. It's always tricky. You have to kind of go from 3D to 2D. And so we're catching just the very bottom of the frontal horn of the lateral ventricle, but then you can see we definitely hit the atrium towards the back. And then this is through the third ventricle. There's third ventricle. One thing about the basal ganglia, so the caudate nucleus is found on the lateral wall of the lateral ventricle, but the head of it actually is, its extent goes down more inferiorly than the frontal horn of the lateral ventricle. So we're going to run out of seeing frontal horn of lateral ventricle, but we're going to keep seeing the head of the caudate nucleus. So here, let's see what we can see for basal ganglia here. So there is our head of the caudate nucleus, exactly where it's supposed to be on the lateral wall of the frontal horn of the lateral ventricle. And then atrium, if I said if there's lateral ventricle and there's caudate back here, see how much smaller the tail of the caudate is on the lateral wall of the lateral ventricle. Then on the other side of the internal capsule, just medial to the insula, we definitely see some putamen. And then here, as we get down a little bit lower, we start to see some of the other deep nuclei of the basal ganglia that are called the globus pallidus. So globus putamen and globus pallidus, those are called the lentiform nuclei because I guess they look like lentils. I try not to think of food when I identify brain structures, but this happens occasionally. So here we can see putamen and then globus pallidus, little different look to it. It's a little bit darker. Neurons doing a little bit different thing than the putamen does. Then we've got this white matter that separates the head of the caudate nucleus from the putamen. At this level, this is the anterior limb of the internal capsule. Then the genu of the internal capsule is adjacent to the interventricular foramen. And then the posterior limb of the internal capsule, lateral to it, we have these deep nuclei of the basal ganglia. Medial to it, we're going to see the thalamus. So before I do my thalamus animation, let's finish up some other circuitry that is associated or adjacent to the basal ganglia. So remember, lateral wall of lateral ventricle, we've got the caudate nucleus, but then just medial to the caudate nucleus is where we have that outflow tract of the amygdala called the stria terminalis. And so here we see stria back here, but then anteriorly, there's also a structure called the bed nucleus of the stria terminalis that we see up front here. So that it actually makes a bump. We can see kind of the bump over here of the bed nucleus of the stria terminalis. All right, so there is all of that, those structures put in on this MRI. So putamen and external globus pallidus, and then the region of the bed nucleus of the stria terminalis. Then I want to stick my thalamus in there. So there's thalamus on either side of the third ventricle. And then what about structures associated with the hippocampus? So here we see at the back, what's called the crus of the fornix, and then we see the columns of the fornix anteriorly here. So here's a three-dimensional structure showing down in the temporal horn of the lateral ventricle is where we find the hippocampal formation. And then the axons coming out from that ascend and form the crus, or both of them together would be the crura. So here we see these things ascending, and then they come anteriorly and then descend, and on a horizontal section, we hit the columns of the fornix as well. Notice on this diagram, we've got posterior parts of the columns of the fornix that are going to synapse in the mammillary bodies, but then we also have precommissural fibers that are actually projecting to both the septal nuclei and then nuclei of the basal forebrain. So these are all cholinergic nuclei that actually are some of the first regions to degenerate in Alzheimer's disease, which I guess to me underscores their importance in memory consolidation. So, all right, so there is where we see column of the fornix and then the septal nuclei. I've also gone ahead and identified insula on this. And then here we see, here would be the crura or the crus of the fornix coming up, column of the fornix as it's descending down, and then regions of the septal nuclei on this image as well. And then I apparently was super excited about the internal cerebral vein. Here's another diagram kind of showing takes a little bit of mind bending to figure out how this fits into this. but hopefully this makes it a little easier to see. We're hitting the cruise of the fornix back here and the columns of the fornix posteriorly and then anteriorly right there. And then this is another specimen that is actually, it is actually this specimen. So what I did is I just kind of tipped it up. And so you see how the columns of the fornix are kind of descending down here, and you can see the structures actually descend down, and this would be post-commissural fibers. So okay, they have to be behind the anterior commissure, and you can see the anterior commissure right there. So when I teach my students, I always say, you want to look, it kind of looks like an A shape, and it's like little snake teeth come down right behind the columns of the fornix, and you get this very stereotypical appearance. This, I think, is also a nice time to review the circuit of Pepez in terms of kind of what structures that we're looking at here. So in terms of how do we consolidate, or how do we turn these moment-to-moment experiences into this thing that we call long-term memory, we have all of this cortical activity that's happening for our moment-to-moment perceptions and thoughts. And those regions are connected to the cingulate gyrus. And then the cingulate gyrus gives rise to some axons come out of it that are called the cingulum that project to this region. So this structure is called the parahippocampal gyrus. It's made of entorhinal cortex. And you'll notice in my little text right here, this shows cingulate gyrus, that's the neuron, and then the axon that comes out of that neuron forms the tract. So we call that the cingulum, and then it synapses on the parahippocampal gyrus. So that's just kind of my way I try and show we're talking a lot of times about, we name the collection of cell bodies one thing, and then we name the axon something else, but they're really all the same structure. And then to get from the parahippocampal gyrus into the hippocampal formation itself, we've got two pathways that we'll look at a little bit later more specifically. Then output, the magic of memory consolidation happens somehow, probably long-term potentiation at synapses in the hippocampal formation, but then we've got to get that information back out because memories are actually stored elsewhere. So the hippocampal formation, the axons that we see of the fornix have arisen from this structure called the hippocampal formation. So, oh, I'm missing a here. This is me throwing a bunch of slides together. So this word shouldn't be there. So the hippocampal formation gives rise to the fornix, and then the post-commissural fibers of the fornix. So there's actually anterior commissure caught in cross-section, project to the mammillary body of the hypothalamus, and then the pre-commissural fibers project to both the septal nuclei and then the nuclei of the basal forebrain. So those are all cholinergic nuclei that have widespread cortical projections that are very important in memory consolidation. In terms of actual circuit of Pepez or Papes circuit, we've got a projection from the mammillothalamic, or from the mammillary body, forms the mammillothalamic tract, projects to the thalamus, both the anterior nucleus and the mediodorsal nuclei of the thalami, and then they project back to where we started from, to the cingulate gyrus via this, just hops a ride on the internal capsule to get back to cortex. And then the cingulate gyrus, once basically we run this circuit, ultimately memories are distributed across the cortex, usually in kind of relevant cortical areas for long-term memory storage. And so this is known as the circuit of Pepez. I like to call it the hamster wheel of memory, just because I think it's funny. And so we have activity in this circuit, and then memories, once they're consolidated, they are stored elsewhere in the cortex. So what we're seeing here, portions of the circuit of Pepez, here we see basically fornix, and then also these septal nuclei are receiving projections from the fornix as well. Okay, so here is a lower down cut. So you'll notice partially we can see we're lower down because we have no frontal horn of lateral ventricle left. And so why I like this section right here is that it actually, I was so proud when I did this, I cut right through the anterior commissure. So you can see, oh, boom, there it is, white matter structure that actually is connecting the temporal lobes. And so here on this MRI, we can also see the anterior commissure. Behind the anterior commissure, we can see these rounded structures where we've caught the columns of the fornix as they descend down, and they actually pass through the hypothalamus. So that is the gray matter around them. Back here, we're catching the cruise of the fornix, so the legs of the fornix are right here, and then this bump is actually the hippocampal formation itself. And then those axons come around and descend as the columns of the fornix. So on the MRI, we can see, oh, there's hippocampal formation back here, associated with the medial wall of the lateral ventricle. And we can actually see, I get very excited when you can see the columns of the fornix descending down behind the anterior commissure. Then anterior to this is where we're going to find the septal nuclei, so those cholinergic nuclei associated with the basal forebrain. So ventricular structures that we can see, we've got a nice third ventricle, and then a nice atrium of the lateral ventricle back here. So here we see our third ventricle, atrium of the lateral ventricle. Lateral to the third ventricle is where we have our thalamus. As we get down a little bit lower, we see some other structures associated with the thalamus. So there's this little triangle of tissue called the habenula. And then the pineal gland kind of hangs off the back of the paired habenuli. And to me, that's always a very notable structure. So here you see the thalamus, and then you can see that little triangle of the habenula and the pineal gland, a nice midline structure hanging off the back. And habenula, I mention it because it's actually really important, involved in outflow of emotion from the amygdala. Notice I did color it purple. So for kind of physical expression of emotion, it is associated with anxiety and depression. So if you do research on that, you probably heard of the habenula. Then step back, we also need to talk about our basal ganglia at this point. So these nuclei at the base of the telencephalon. So we've got telencephalon, 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 and then there are nuclei at the base of the telencephalon. So what can we see at this level? Well, like I said, head of caudate actually descends down below the frontal horn of the lateral ventricle. So we do have some head of caudate. And I'm going a little bit out on a limb here, but I do think that might actually be a little bit of nucleus accumbens, which all of us who work with psychiatric type things, nucleus accumbens is incredibly important in reward and is implicated in addiction, which is definitely something we as a population deal with. Then lateral to kind of this internal white matter is the putamen. So notice it's going to kind of squeezed in between the insula and then this white matter of the internal capsule. And then at this level, as you get down even lower, so up above, we just saw one part of the globus pallidus. We saw just this external globus pallidus. At this level, we also are getting down low enough we can see the internal globus pallidus as well. So it's definitely the smallest and the deepest and the most medial of the lentiform or the lentil-shaped nuclei. So have put this corresponding structures in on the MRI. And then also at this level, I believe this is the bed nucleus of the stria terminalis. So we've got stria terminalis back here, and then bed nucleus of stria terminalis kind of adjacent to the anterior commissure. There's not much for anterior limb of internal capsule, but we definitely can see some anterior limb of internal capsule separating the head of the caudate from the putamen. We can see the posterior limb of the internal capsule separating the lentiform nuclei from the thalamus. And then there we go, there's some insula. Then I don't have a corresponding MRI with this, but this is actually a section that's essentially, it may actually be the bottom of this slice. I maybe just flipped it over. So you can see, we still are seeing these three lentiform nuclei. So the putamen, the external and the internal globus pallidus. And this section caught the anterior commissure kind of right through the middle. But here we're seeing, so on the other side, we're seeing the top of this. Here we're seeing kind of a bottom of this. It has a little bit of like a wavy M shape. So that is just why we're seeing what we're seeing here. Then medial to these lentiform nuclei, we've got the posterior limb of the internal capsule. This level, I terrify medical students or PA students when I say, oh, guess what you can see here? There's actually, I see the subthalamic nucleus right there, which is part of basal ganglia circuitry and actually a really important part of internal circuitry for braking activation of the cortex. Then here we see the subthalamic nucleus. There's actually this little triangle of the habenula is right there. And then we're seeing the pineal gland as well. In terms of circuitry associated with the hippocampal formation, here we've got this kind of back part, the hippocampal. Basically, I don't have a good cross-section with the hippocampal formation on axial section because essentially what happens is my brain falls apart by the time I get down low enough to cut that. So I just kind of see these back parts of the hippocampal formation as it's kind of coming up there. And then here is the white matter of the fornix. And then we can see this region is the hypothalamus. And in here, I actually can see the column of the fornix as it's descending through the hypothalamus to the mammillary bodies. Septal nuclei would be in this region. So, and then also I'm going to show you a different picture of this same section. Again, things I get excited about. So the columns of the fornix synapse in the mammillary body, and then the mammillary body projects up to the anterior nucleus of the thalamus. And so we can actually see the mammillothalamic tract, that projection on this cross section. So if I zoom in a little bit, there are columns of the fornix, and then right in here, we can kind of see it over here. Maybe put on, I always call it your histology imagination. Maybe it's your neuroanatomy imaginations, but that would be the mammillothalamic tract as it's ascending from the hypothalamus. And then these two sections are down a little bit lower, and they are through the midbrain. So if we identify there is, or I guess we're below the frontal horn of the lateral ventricle, we see some atrium of the lateral ventricle, and then we can see this midbrain. So there's midbrain and midbrain up a little bit higher. This space, instead of being, this is actually third ventricle, the very bottom of the third ventricle. So up higher on either side of the third ventricle is where we see the thalamus, but then as we get down to these lower sections, we see the hypothalamus on either side of the third ventricle. And really all the third ventricle is a slit-like space between the diencephalon, so between the thalamus and the hypothalamus. So it's just a space between those structures. Things I see in the midbrain, this is up a little bit higher versus a little bit lower. At this level, I actually see a really nice subthalamic nucleus, which again is, I've forgotten, didn't make it orange on the other slide, but I put it orange on here because it is associated with basal ganglia circuitry. Also, this one, we see some really nice substantia nigra on this side. This side, I'm curious if this person didn't have a little bit of Parkinsonism. So substantia nigra, literally called substance black, clever neuroanatomist naming. But if you recall, this structure, incredibly important in releasing dopamine onto the nuclei of the basal ganglia, onto the striatum. So the caudate, putamen, nucleus accumbens, and that is incredibly necessary goose to the system to allow our cortex to work properly. And then of course those dopaminergic connections are essentially what are hijacked by illicit drugs to have, we just have abnormal reward seeking where all we do is seek the drug instead of, the good things that we should seek in life. All right, so I said this was the third ventricle. So on either side of the third ventricle, we should see the hypothalamus. Again, things I get excited about. So there's hypothalamus. Here I can actually see the white matter of the columns of the fornix synapsing in the mammillary bodies. It's there, I swear to you. Over here, we can see we're a little bit lower down. So we see the hypothalamus, and then we can just see the mammillary body proper itself there. In terms of structures associated with kind of the medial part of the lateral ventricle. So the way I teach my students, caudate nucleus is lateral wall of lateral ventricle. For you guys, because it's actually a notable structure, the stria terminalis, which is outflow from the amygdala, is just adjacent to the caudate nucleus. Then in terms of relative location of the hippocampal formation, that structure is on the medial wall of the temporal horn and the atrium of the lateral ventricle. So if we look medially here, we see kind of this little swirly shape that is the hippocampal formation. And then we can see the white matter that's called the fimbria. The fimbria of the fornix form, they, again, like, if you want to define it, we've got the alveus is when the axons coming out of the hippocampus are stuck to the back of the fornix. And then as they start to, they detach a little bit and they're called the fimbria. And then as they start to go up, they're called the legs or the crura, and then the body, and then the columns of the fornix as they come down. So all the same axon, just different names depending on where it is. Then lateral wall of the lateral ventricle, here we can see caudate and then stria terminalis. So over here is also caudate and stria terminalis. And then, oh, okay, so I labeled the pineal gland. Also, if you're curious, this stuff right here that's just adjacent to the midbrain, those are really low down nuclei of the thalamus. So the pulvinar nucleus, I always call the butt of the thalamus, kind of hangs over the back adjacent to or kind of on either side of the pineal gland. And then here we're actually seeing medial geniculate nucleus and lateral geniculate nucleus, which these are the thalamic relays. Medial music for auditory relay, lateral geniculate nucleus for light, so for visual relay from the retina. And then we do have some nuclei of the basal ganglia here. I'd say, yeah, that's probably caudate, maybe some nucleus accumbens, some putamen out there, not as easy to identify at this level. There's some insula, and then this is similar. This, I would say this MRI is kind of in between those two levels. But here I see midbrain, and then this would be these regions of the hypothalamus. And here we can see the hippocampal formation and then the atrium of the lateral ventricle. And then insula would be out there. And then I guess I just got excited and showed you just, here's another cut from a different brain where we can see frontal horn of the lateral ventricle, the atrium of the lateral ventricle. We can actually see the caudate, or sorry, the putamen and the external and the internal globus pallidus a little bit better. There's the subthalamic nucleus. There's a little bit, which is so degenerated. I always hate it when I chop up a brain and the holes are so big. But we can see the anterior commissure. This would be the bed nucleus of the stria terminalis with the the septal nuclei of the basal forebrain there, and then the column of the fornix as it's descending down. And then there's the hippocampal formation itself in the back. So that was horizontal sections. So now I'm going to take you through some coronal sections. Like I said, lots of anatomy here. So always want to orient. Axial sections have this oval shape to them, where if you're up through the cortex, you should see cortex all the way around. Coronal sections have more of kind of like a muffin top shape or like a mushroomy type shape. And if they have a stalk down here, it will be the, excuse me, it will be the brainstem. So this coronal section, when I teach the anatomy of the brain, I start with this coronal section because kind of in front of here, the brain isn't, the two hemispheres aren't connected together, so they just kind of fall apart. Of course, you'll see it on MRI or imaging, but here is kind of where we have our first notable structures. So this level I make my students look at because this is the level of the nucleus accumbens, that super, super important structure. So here, always good to start with the ventricle. So here we see this would be the frontal horn of the lateral ventricle. There's frontal horn and lateral ventricle. Then lateral wall of lateral ventricle, always and forever, we'll see the caudate nucleus. So there's the head of the caudate nucleus. Then lateral to the internal capsule, we'll see the putamen. And then at this level, we have the nucleus accumbens joining these two regions. This is anterior limb of internal capsule. And I always call this, looks like the sad broken heart level of the basal ganglia. And why are they sad? Well, because these nuclei actually would love to be continuous. The reason they're called the striatum is because there are liney, stria, cell bridges connecting the head of the caudate and the putamen, but the internal capsule goes right through the middle of them and keeps them separate forever. So that is very sad in my head. I don't know. I enjoy neuroanatomy. So here is these same structures shown on this corresponding MRI. This is the septum pellucidum. So down at the base, we'll have septal nuclei. And then these actually are precommissural fibers of the fornix that are projecting to the septal nuclei in the basal forebrain. And if we look over here, we can kind of see nuclei and then some fibers, some axons right there. This level is also defined by the optic chiasm, and then we can actually see a little bit of the pituitary stalk, the infundibulum coming down from there. So there's the optic chiasm, there is the infundibulum, and actually here's some pituitary right there. Temporal lobe wise, we want to look at amygdala and hippocampal formation in the temporal lobe. At this point, we see pretty normal thickness of cortex, and so we're not quite to the amygdala at this point. So this is our next level back that I look at with my students. So kind of my canonical coronal section. So again, you want to orient. This is a coronal section, so it doesn't have that oval shape. It's got kind of that muffiny mushroom, head of a mushroom shape. It does still have a nice ventricle in there. So there is a frontal horn of the lateral ventricle. Here we see frontal horn of the lateral ventricle. Found on the lateral wall of the lateral ventricle is the caudate nucleus. This far forward, it's going to be pretty big. It will be the head of the caudate nucleus. Then on the other side of the internal capsule is the putamen. And then here we're getting far enough back, we can actually see external globus pallidus. So there is external globus pallidus. And here are these structures on this corresponding MRI. This is also a level I look at because when I do it right, I hit the anterior commissure. So if we back it up, you see it's kind of got a little swervy M shape. So I get three different parts of it here. If you look over here, though, there is actually anterior commissure on this MRI. And so associated kind of with these anterior regions, we should see this is septal nuclei. These are actually precommissural fibers of the fornix that they're going in front of the anterior commissure to the septal nuclei in the basal forebrain. And then here we see the bed nucleus of the stria terminalis. So it's definitely more of a nucleus than just this outflow structure. So here are these structures over here. This midline space then is the third ventricle. On either side of the third ventricle is the hypothalamus, and we actually can see some infundibulum right there. So here's hypothalamus and a little bit of infundibulum right there. And then this level right of the brain, right under the anterior commissure, is where we see the cholinergic nuclei of the basal forebrain. So this is actually nucleus basalis of Maynard, and this is called the diagonal band of Broca. I believe this may be called the something innominata. I don't know. That's not a name I know it from. So, but this is where these structures are on this corresponding MRI. So now we're getting some exciting things happening in the temporal lobe. So if we look down here, we have a little bump that is the structure called the uncus. The uncus ends up being clinically important because of basically the anatomy of the cranial vault. If we've got like an epidural hematoma, we have some kind of bleed in the brain, it will push the brain down, and the uncus is the part that herniates over a structure called the cerebellar tentorium and compresses the brainstem and causes all sorts of trouble. Anatomically, though, it is under this bump is where we find the nucleus, or the three nuclei that make up the structure called the amygdala. So here we see lovely amygdala over here. It's not quite as perfect on this side, although we can see some uncus right there. So there is uncus. We see this bump of the uncus, and then this nucleoid structure, the amygdala on there. The amygdala actually has three different nuclei in it. The basolateral nucleus is important in inward emotional responses. So this is going to have a lot of projections internally. The central nucleus is for outward physical expression of emotion. So there's going to be projections essentially to the brainstem. And then the corticomedial nucleus is associated with olfaction and appetite. On this section, this region right here is actually defined as one of these outflow tracts from the basolateral amygdala called the uncinate fasciculus. And then remember that the stria terminalis is also outflow from the amygdala. So it's actually coming around, it's following the C shape of the lateral ventricle to get to the front of the brain. Versus the uncinate fasciculus is more of kind of like a direct route to get to the front of the brain. Okay, this next section back is a section through the genu of the internal capsule. So also in terms of initial orienting, this is a coronal section. So we do have some lateral ventricle. Technically at this point, I would call this body of the lateral ventricle. I always tell my students, no, I won't tell you to be specific at this level because it does look kind of big still. And then lateral wall of lateral ventricle, we still have the caudate. At this point, I would call this the body of the caudate. The stria terminalis is always, again, that outflow from the amygdala, always medial to the caudate. We've got the putamen out here laterally. And then now that we're further back, we see all three nuclei of the lentiform nuclei. So we see the external and the internal globus pallidus as well. I always think the genu of the internal capsule looks kind of messy. It's not as discrete as the anterior limb and the posterior limb. In part, that's because there's actually anterior thalamic nuclei that are kind of protruding into this region. And then another marker for genu of internal capsule is this structure right here, which I call everything structure for my students, but this is interventricular foramen. So the actual opening between the lateral ventricle and the third ventricle. And then at the front of the interventricular foramen, we can actually see those columns of the fornix as they come down, and we can see the anterior commissure on the other side. So this would be body of the fornix as we cut it in cross section, and then we can see the columns of the fornix. And then remember, these structures are going down to synapse in the mammillary bodies. Another structure that's characteristic of this level are the mammillary bodies of the hypothalamus. So we can see the white matter of the columns of the fornix projecting to the mammillary bodies of the hypothalamus. And so this region then would be the hypothalamus. So if we look over here, it's all there. It's really there. We can see there is cross-section body of the caudate. And then there are the columns of the fornix as they come down. This is the interventricular foramen. And then down here, we can see the little round balls of the mammillary bodies. And I actually see some dark matter on a T2 MRI, white matter actually shows up dark of the columns of the fornix as they're synapsing in the mammillary bodies. Then this region would be the hypothalamus. We also still are seeing some uncus. And underneath the uncus is the amygdala, but here we're getting far enough back that we're actually also seeing some temporal horn of the lateral ventricle, which is ginormous in this old person's brain. They have some problems going on here. We see some neurodegeneration. But once you start to see the temporal horn of the lateral ventricle down on these coronal sections, on the medial wall of that structure is the hippocampal formation. So whenever you see lateral ventricle, you should always see some sort of caudate on the lateral wall, medial wall of the temporal horn, and the atrium of the lateral ventricle. That's hippocampal formation. So here we can see the uncus, and then this would be amygdala. So kind of more nucleoid, kind of homogeneous structure here. And then here we see some temporal horn of the lateral ventricle. That makes this the hippocampal formation. And so when I talk to students to kind of identify the difference, I always say the amygdala looks, it's very homogeneous versus, I don't know if you guys went through a phase in like third grade when you were like a girl and or maybe you weren't, some of you probably were never girls. But I went through this phase where you'd like outline things and then shade kind of in, like I still have a whole Precious Moments coloring book that my sister's still mad at me that I wouldn't let her color in, and I probably still wouldn't let her color in, that has that kind of shading. Well, if you look at the hippocampal formation, you see kind of that same kind of line shading internal structure to that, which has to do with the fact that it's actually some three-layered cortex that got folded in itself. And so that is how you can differentiate the amygdala from the hippocampal formation. This is a slice at the same level from a different brain. So you can kind of compare and contrast, see these same things. So here is lateral ventricle, Here we see the nuclei of the basal ganglia. And again, we can see all three of these lentiform nuclei. There is lovely stria terminalis. And then here we can see the previous section, we really just saw the body of the caudate, but here we can actually see there's definitely some septal nuclei there. There's the mammillary bodies. This specimen is very exciting because you can see both the columns of the fornix projecting to the mammillary bodies and then the mammillothalamic tract coming out of that structure. So column of the fornix, mammillothalamic tract. In the temporal lobe, on this side, we're pretty much past amygdala, but on this side, we still have a little bit of amygdala. And then on the medial wall of the temporal horn of the lateral ventricle, we can see this kind of lininess of the hippocampal formation. And so then these coronal sections are through the posterior limb of the internal capsule, and they're characterized by the thalamus. So again, always good to start with the whole. So here's the body of the lateral ventricle and the temporal horn of the lateral ventricle. And then we've got the third ventricle right here. In terms of basal ganglia, we've got body of the caudate, stria terminalis, and actually you can see the whole of the thalamostriate vein. We have the putamen and both the external and the internal globus pallidus. Down here, we've got tail of the caudate, and then it would be easier to tell in a cell stain, but this region would be stria terminalis. This is the posterior limb of the internal capsule. Why? Because we've got thalamus medially. So our other sections didn't see thalamus on the medial surface. We saw some hypothalamus, but not thalamus. So once we get to the point of the thalamus, that makes this the posterior limb of the internal capsule. This level I show my students because here we've got nice subthalamic nucleus. And then let's see, then down here in temporal lobe, we can see the hippocampal formation. This brain makes me sad because it's perfect in every respect, except the hippocampus is just kind of bad things were happening to this person's brain. And then here is kind of it's called the fimbria of the fornix coming off of the hippocampal formation. And then here we see the body of the fornix there. This section I also show students because it catches the mammillothalamic tract as it goes through the thalamus to synapse in the anterior nucleus of the thalamus. Over here, we see similar ventricular structures with the body of the lateral ventricle, the third ventricle, and then the temporal horn of the lateral ventricle. Here though, we're back about, I don't know, about that far, and we've suddenly we're running out of basal ganglia. So you look out lateral to the internal capsule between the insula and the internal capsule. There's just a little bit of putamen left. So we're past the point where we've got the external and the internal globus pallidus. So here's posterior limb of the internal capsule. Here is the thalamus. Here is we got a little bit of substantia nigra here, but here we have really nice substantia nigra. Again, that is the structures that degenerate in Parkinson's disease and then the Parkinsonisms. We also have a loss of the substantia nigra. This side makes me so sad. Look at that horrifically degenerated hippocampal formation. And then we can see the body of the fornix right there as well. So here's a corresponding MRI that is at about this level. Notice how tiny the third ventricle is in a young person's brain, how tiny the temporal horn of the lateral ventricle is in a young person's brain. Here's where we see caudate putamen. And then I'd say this has a little bit of globus pallidus here as well. Here we see the thalamus. This would be the posterior limb of the internal capsule. Down here in the temporal lobe, we've got hippocampal formation. And then I was going to look at a couple parasagittal sections, and then we'll look at some 3D. I'm basically not going to be able to get to most of this talk. So I think I knew that in my heart, but anatomy, we're doing anatomy. So this parasagittal section, these are not as commonly seen, but in terms of seeing the hippocampal formation, this really is actually one of the best ways to do it. So here we're seeing the insula deep in the lateral fissure. This would be temporal lobe, and we're seeing just a little bit of the hippocampal formation and a little bit of temporal horn of lateral ventricle right there. Then there'd be parietal occipital lobe. Here is a stained parasagittal section, and look at how beautiful, we can see the lininess of the hippocampal formation. So I don't know if it looks like a seahorse, but it is, I don't know, it is a really lovely structure. And then in front of it, we see the amygdala. And so you can see how on coronal section, you could have a section where you would just hit the amygdala, and then section when you would hit the amygdala and the hippocampal formation. And then you get back behind the amygdala. So here is moving out a little bit more laterally. So the insula is not quite as obvious. Here, though, we can see this is the most anterior tip of the temporal horn of the lateral ventricle. We've got the atrium of the lateral ventricle back here. And then here is, if I back this up, you can see there is the hippocampal formation, just right exactly where it should be and shaped the way it should be. And then as we move out even more laterally, I would say this would be region of the amygdala right here. And actually back here, there's probably some amygdala right there as well. And then a little bit of temporal horn of lateral ventricle, and actually here would be some hippocampal formation right here. And this would be a crus of the fornix as the legs, the crura are coming up, and then some atrium of the lateral ventricle. In terms of basal ganglia, I believe it was Dr. Cotton talked about, or maybe it was Dr. Coltis, I can't remember. Somebody mentioned how the basal ganglia can be kind of like an ice cream cone shape. And so I agree with that. I once taught that. And so kind of how these structures relate. Like I said, the caudate nucleus is a C-shaped structure found on the lateral wall of the lateral ventricle. And notice the head of the caudate definitely descends down below the frontal horn of the lateral ventricle. So here we see some stained sections, which this was a blast from my past, but here we see head of the caudate, and then the body of the caudate then is above the plane of section in all of these axial sections that we looked at. And then around disc-shaped structures are the putamen, the external and the internal globus pallidus. So they, of course, are lateral to the internal capsule versus the caudate is medial to the internal capsule. So the putamen is this big one, and notice on staining, it does look identical in consistency to the caudate nucleus. And then as we go more medially, and they get smaller from lateral to medial, are the external and the internal globus pallidus. So when we look at these in coronal section, then we can see a little bit different appearance. Always though, on the lateral wall of the lateral ventricle, hopefully you picked that up from me at this point, we will see caudate. And then as we progress back, of course, head of the caudate is going to be bigger than body of the caudate. And then these disc-shaped structures lateral to the internal capsule, we've got the putamen and the globus pallidus, and then nucleus accumbens most anterior. So in terms of this ice cream cone shape, I used to like hold sticks and like have a whole bunch of slices laid out, and like I probably looked like a crazy person talking to my medical students, but it's very exciting. So the ice cream cone shape of the putamen and the external globus pallidus. So putamen is like the ice cream, and then the external globus pallidus is kind of like the chocolate dipped part of the cone, and then the GPI is that perfect little bottom part of the cone. So if we make our little cone right here, and then we have to rotate it a little bit, because in terms of analogies, I also think the putamen is kind of like Princess Leia buns, and then turn that into ice cream and orient it where the cone is down a little bit. That is the shape of these structures. So here we see what I'm trying to convey. And so on an axial or horizontal section, here we see the putamen and the external and the internal globus pallidus. So if we cut up higher, you can see how you would just see external globus pallidus and putamen, or you would just be able to see putamen on these various sections. And then in coronal section, though, I would say no matter which way you slice or dice the brain, these structures are going to be present anatomically relative to each other in the same orientation, no matter if it's a coronal section or horizontal section or a sagittal section. So on coronal section, you can also see how we would see putamen, external and internal globus pallidus. So again, more anterior cuts. So here, this section through the nucleus accumbens, we're just seeing the putamen versus this section that is hitting the anterior commissure, we actually are seeing both putamen and external and internal globus pallidus. To see all of the parts of the lentiform nuclei, so to include the internal globus pallidus, we will see that on cuts basically kind of right behind or at the level of the anterior commissure back to anterior regions of the thalamus. So just a couple slices will show that up. Okay, so that was all the anatomy questions. So I don't know if we're going to get to much. We might just do some selections from the other slides. But if you guys have some questions, let's go ahead and answer a few of those.
That was a lovely overview, a very detailed overview of neuroanatomy. We have some excitement about your slides, and just letting everyone know that those will be uploaded later this week and that you have offered to provide a PDF too. So those will be coming. So thank you so much for that. But I think I'll keep on monitoring questions, but otherwise I think we're okay to move forward.
Okay, cool. All right. Well, let's go ahead and talk through some neural circuitry of the basal ganglia. So all of these structures have input into them, and then they do their processing, and then they have output to affect either changes in cortical function or essentially changes in the periphery through connections to the brainstem and the spinal cord. So most of cerebral cortex, kind of anterior cerebral cortex, and then the hippocampus and the amygdala all talk to the basal ganglia. And then there is critical modulation by the substantia nigra in the ventral tegmental area. So that dopamine release is a big, big deal. So we have a mesostriatal pathway to the caudate and the putamen. We have a mesolimbic pathway from the ventral tegmental area to nucleus accumbens we see here, and then also to this limbic lobe that's critical in reward and addiction. And then we also have projections from both the substantia nigra and this ventral tegmental area, just medial part of the midbrain, to regulate function of the prefrontal cortex. So this is for executive function, also our affect, like how we present ourselves, and then working memory as well. And so Dr. Cotton had some great cases showing individuals who had degeneration of the substantia nigra, but with some slightly different manifestations. So degeneration that is primarily affecting the mesostriatal pathway will just present with motor symptoms, versus there was a man who had both motor symptoms, but also problems with this mesolimbic pathway, who just had very abnormal mood. And then also a man who had difficulty essentially with kind of prefrontal cortex function. So he was slowed mentally, and then a flat affect as well. So internal circuitry of the basal ganglia, Dr. Cotton also went over this. We have all of that input from cerebral cortex into the striatum, which striatum refers to caudate, putamen, also I would put nucleus accumbens in there as well. And we have a direct pathway that is output to the thalamus that then essentially gooses the cerebral cortex, that is for facilitation of neuronal activity in cortex. And then we have this indirect pathway that's a little more complicated, that its effect is essentially a brake on cortical activity. So dopamine release from the substantia nigra activates the direct pathway, and it inhibits the indirect pathway. And again, how does that happen? Because we've got different dopaminergic receptors on different regions of the striatum, or different neurons in the striatum. And then also important to note is there are cholinergic interneurons in the striatum, again, caudate, putamen, that seem to be more active in the indirect pathway. So outflow from the basal ganglia, again, this is just what they got to get out to cause an effect. So outflow from the putamen is primarily for activating motor cortex. So problems with this function will give us the dyskinesias, the akinesia, or hypokinesia, hyperkinesia, rigidity, tremor. Body of the caudate actually gates frontal eye field. So for Huntington disease, Some of the first manifestations are actually slowed saccadic eye movements because of dysfunction of this channel through the basal ganglia. The head of the caudate is for gating prefrontal cortex. Again, Huntington's disease is where we really see altered behavior because that is specifically degeneration of the head of the caudate. And then the ventral striatum, so the output from the nucleus accumbens, and that includes just the little bit of caudate and the little bit of putamen adjacent to that. That's for control of these limbic cortices. And again, that will manifest with abnormal emotions and also changes in learning and memory. Pharmacotherapy of basal ganglia disorders, if you kind of know, like the medical students and the PA students, knowing this circuitry is knowing the rationale for the drugs that we use. So for the Parkinsonian, or basically Parkinson's disease, not the Parkinsonisms, the big goal is we lost striatal dopamine. So from the substantia nigra, so we need to supplement striatal dopamine. So dopamine replacement is the classic, but then there are also dopamine agonists and then drugs that inhibit metabolism of dopamine. Also very important, and I'm sure you guys have run into your practice at some point, is all of the antipsychotics, the neuroleptics, have antidopaminergic activity. And on that, when those drugs are in the brain, the striatum can be like, wait, I need my dopamine. And so they'll actually upregulate their dopamine receptors. And you end up with this increased activity that is manifests as a disorder called tardive dyskinesia. So delayed onset of bad movements that, unfortunately, the problem with that is that sticks around even after you take the medication away. Other medications, so anticholinergic medications are actually helpful in relieving tremor and used for acute dystonia. So they are presumably working within this part of the striatum. And then Dr. Cotton also talked about deep brain stimulation. So, and one thing I'm not sure if she noted or not that I really had to struggle with was they call it stimulation. It's not stimulation. It is knocking out electrical function. So it's kind of like a temporary lesion. So knocking out function of the subthalamic nucleus is actually really helpful in relieving both bradykinesia and then also the resting tremor of Parkinson's disease. Placement of a stimulator, which again is knocking out function in the internal globus pallidus, reduces that inhibitory output from the basal ganglia, and that is actually particularly helpful with rigidity and also can be used to treat dystonia. And then placement of DBS placement in the thalamus, specifically the ventrolateral nucleus. Well, actually, one of the more common movement disorders is essential tremor, and so that helps with essential tremor if that's causing problems with movement. If we have if we want to decrease the amount of dopamine present activating the striatum, which this of course would be Huntington's disease, this when you're decreasing dopamine, of course, you're going to have mood problems. So all of these medications have a side effect of depression, which can manifest as suicidality. And so you tend to kind of try and stepwise, let's inhibit reuptake of dopamine. And then let's try the atypical antipsychotics that don't have as strong of antidopaminergic action. And then finally move to the typical antipsychotics that have much more strong antidopaminergic activity. And then in terms of treating basal ganglia disorders peripherally, sometimes it's like, oh, we don't even want to get into this hot mess. So let's just go ahead and prevent the muscles from actually contracting with botulinum toxin at the neuromuscular junction. So hyperkinetic disorders of the basal ganglia. Hemiballismus is a disorder of the subthalamic nucleus that manifests with unilateral flinging and flapping, usually arm more so than leg. And I think this is so clever. Remember that placing a stimulator, which again knocks out function in the subthalamic nucleus, is actually on a background where you have not enough movement. If you then inhibit the subthalamic nucleus, that'll move you up to more normal levels of movement. Wilson disease is a disorder that is a genetic disorder of copper excretion. And so we'll look at an MRI that shows basically you have copper accumulation in your basal ganglia, and it also produces a hyperkinesia. and then Kayser-Fleischer rings in the cornea. Huntington disease is kind of the flagship hyperkinetic disorder where we've got a triplet repeat problem in this gene for Huntington protein, and it causes progressive degeneration of the striatum starting in the head of the caudate. And so that actually manifests as initially actually psychotic movement issues, but then behavioral changes end up being a pretty big deal early on in the disorder. Dystonia, lots of kinds of dystonia that people have to deal with that are problems with basal ganglia function. So deep brain stimulation in the internal globus pallidus can be used to relieve dystonia. Essential tremor is actually the most common movement disorder. This one is related to the basal ganglia, maybe not as directly, but there are some medications to treat that. These are side effects of antipsychotics, so messing with that dopaminergic system in your brain. Tics and Tourette's also are, tics, Tourette's, and obsessive compulsive disorder are all kind of tied together, overlapping problems with the basal ganglia. And so there's actually, for OCD, there are some surgical interventions that can help with OCD and that disrupt the connections between the basal ganglia and the cortex. So here, hopefully this MRI is a little more kind of know a little bit more what you're looking at. Now that we've done the anatomy here, we can see the head of the caudate and then the putamen, and they should not be bright like this. Also, this is because of copper deposits. And then there is that Kayser-Fleischer ring in the cornea. Here we see an individual with Huntington disease, and oh man, there is not much for head of caudate here, and there's actually not much for putamen either. This is a nice diagram showing Tourette syndrome, and that really is kind of some overlap with ADHD and OCD. And then OCD, I essentially think of as you've got just kind of too much outward focus. You've got too much do it, not enough emotional control, way too much focused attention on things, just excess outward focus on external environment. So in terms of places where we can disrupt the basal ganglia, like over-activating those regions of cortex, lesion anterior cingulotomy seems to help. And then also, anterior, not cingulotomy, but of the anterior internal capsule, and then also cingulotomy disrupts that connection between the basal ganglia and the cortex. Hypokinetic disorders of the basal ganglia, Parkinson's disease is, of course, degeneration of the substantia nigra. And again, we do have places surgically where we can try and relieve that bradykinesia. Lewy body dementia is also a Parkinsonian disorder. What is important to note about this, though, not only is there degeneration of the substantia nigra, but also of the striatum, so the target neurons. And this is really important to note because this disorder then is going to be insensitive to dopamine replacement therapy because the target neurons, the striatum, has also degenerated. Another tricky thing about Lewy body dementia is, do they have psychosis? Do we need to give them some antipsychotics? Well, no, because they already are low on dopamine. And so giving them something that will even decrease their dopamine levels even further can actually cause permanent hypokinesia, so permanent movement disorders. So this, I think it's important to note, this is a pretty common cause of dementia. Multiple system atrophy there's actually a couple different versions of these that have some different degenerations, but big picture is that they're all insensitive to dopamine replacement therapy because we lose both the substantia nigra and the striatum. And then of note, REM sleep behavior disorder is strongly linked to development of these three. They're called synucleinopathies. And then one of the more common Parkinsonisms that is actually a tauopathy, so it's a different etiology, more like Alzheimer's disease. This, you end up with degeneration at the midbrain diencephalic junction. So lose your substantia nigra and your striatum, can't treat with dopamine, but usually this ends up causing some vertical gaze impairment that manifests. So that is basal ganglia. Neural circuitry of the hippocampal formation. This is the most beautiful stained picture of the hippocampus ever. So terminology, you'll see hippocampus a lot. Technically, it's probably hippocampal formation that is the dentate gyrus with the hippocampus itself, and then also this region called the subiculum. So if you do research, of course, you'll run into CA1 and CA3 regions of the hippocampus pretty frequently. But this is what we're referring to when we talk about hippocampal formation. And sorry, not sorry, Dr. Jenna, is not a corn dog. It is a beautiful swan shape. That is what I see. And then this is the parahippocampal gyrus externally is made up of entorhinal cortex. And this is essentially how you get into and out of the hippocampal formation. So there are two pathways into the hippocampal formation. So there's a perforant pathway that perforates straight into the dentate gyrus. And then there's an alveolar pathway that follows this curve of the alveus into the hippocampal formation. So how do you make declarative memories? Couple things. You've got to, one, you have to be conscious. So hopefully you're all still conscious. And then you also have to have the proper motivation and attention. So there's an executive component to this that your prefrontal cortex and your anterior cingulate cortex is involved in. And then for the consolidation itself, it's those medial temporal and medial diencephalic structures that I've already talked through and have noted that role of the basal forebrain. So the cholinergic nuclei of the septal nuclei, and then nucleus basalis of Maynard, the diagonal band of Broca. Mammillothalamic tract to the thalamus, and then thalamus back to the cingulate gyrus. And again, I like to call this the hamster wheel of memory. We consolidate here, and then storage is in a network across cortex. So if you have someone who's having problems with memory consolidation, it could actually be due to the fact that their prefrontal cortex they have improper attention and motivation that can cause anterograde amnesia. I always say my older son doesn't listen, and so we'll have a whole conversation with all the rest of the children. I'll have explained something, and he was in the room but not attending, and so he will basically ask me the question I just explained because he wasn't catching the hamster. So you got to be able to catch your hamster. And then if you have problems within the circuit of Pepez itself, that will cause anterograde amnesia. And then if we've got problems, kind of strokes out here or dysfunction out here, if we break our storage facility, that will cause loss of remote memories. So disorders producing anterograde amnesia. I thought Dr. Jennett might really enjoy this. This is a reconstruction of HM's brain where he had the horrific seizures. So they thought, oh, let's just take out the medial temporal lobes. And they did. And he woke up, seizures were better, but severe anterograde amnesia. Alzheimer's disease is also kind of the flagship of anterograde amnesia, inability to consolidate. So here's a normal brain, and we can see really lovely hippocampal formations down here, medial temporal lobe, pretty good looking cortex. Here we see Alzheimer's brain, which that looks similar to my section that I showed you with a terribly degenerated hippocampal formation, giant temporal horn of the lateral ventricle. And then also we're going to have atrophy across the cortex as well. Not much in terms of treating this, but cholinesterase inhibitors are used because we also have degeneration in those cholinergic nuclei of the basal forebrain. And so that can help a little bit with cognition early on. It doesn't really delay anything. And then there was some hope for NMDA receptor antagonists, so this thing called memantine, because they think what's happening is these NMDA glutamate receptors are kind of just get cytotoxic. And so maybe if we can inhibit that, but guess what? When you give NMDA receptor antagonists, you can also make somebody act like they're schizophrenic. So kind of problematic in terms of pharmacology. Global hypoxia will also attack the large pyramidal hippocampal neurons. So here's my lovely swan. Here's a lovely swan. And here is, oh, sorrow. What happens after hypoxia? Ischemia. Transient global ischemia. This is a weird one that happens in older individuals where they just have sudden temporary anterograde amnesia that there's just nothing that has caused it, and it actually improves gradually without intervention, maximum about a day. So one of my former students, that she's the family medicine doctor, her mom, this happened. It was quite terrifying. So lots of things can cause memory issues. All right, so neural circuitry of the hypothalamus. So I think what I'm going to just do here, because I only have a couple more minutes, so let's just kind of look at these without paging through it. We've got a lot of input into the hypothalamus. So from your limbic system, basal forebrain, from your retina, amygdala is actually what I want to talk about more than anything. Into the hypothalamus, and then I'm just going to leave these for you guys. Output for the hypothalamus to the pituitary gland. We talked about circuit of Pepez, so other individuals that will have anterograde amnesia. There's this patient NA who got poked up the nose with a fencing foil, and his mammillary bodies got destroyed, and he also has severe anterograde amnesia. Wernicke encephalopathy due to basically an altered nutritional state, not getting your thiamine. Look at our glowing, bright glowing mammillary bodies there, but also degeneration in the medial diencephalon and the fornix. So these individuals will have amnesia, but they also end up with some brainstem problems, so ocular motor dysfunction and ataxia, and then they tend to confabulate as well. So this, just because I am excessive, hypothalamus also influences sleep wake, influences hunger, influences thermoregulation. So again, these slides will be available to you. The last thing I want to note, though, is neural circuitry of the amygdala. So we have all sorts of sensory input into the amygdala. Then our prefrontal, our basically prefrontal cortex also talks to the amygdala. Memory talks to the amygdala, and the amygdala talks to itself. And then we have outward projections from the basolateral amygdala to cortex and subcortex. So to lots of reciprocal connections to the hippocampal formation. And then that uncinate fasciculus is the projection to the prefrontal cortex and the anterior cingulate cortex. And then essentially we have a ventral amygdalofugal pathway, which literally means run away from the amygdala. And so to the brainstem and also to the cortex. But pretty much all of this upward, inward outflow from the amygdala is to essentially talk to both prefrontal cortex and anterior cingulate via the nucleus accumbens and the thalamus. And this is to give us an emotional context for our memory, and then to influence the executive function, basically, how are we going to interface with this environment? Then we have to have an outward physical response as well. So that is, stria terminalis is part of that. So we saw the stria terminalis on a lot of those sections. And then bed nucleus of stria terminalis is associated with anxiety. We also have a projection to the septal nuclei, and then the septal nuclei give rise to this little tract called the stria medullaris, that's what talks to the habenula, and the habenula talks to the brainstem as well. Then that ventral amygdalofugal pathway has internal projections, but it will also join up with the medial forebrain bundle to talk to the brainstem as well. And pretty much all of the diffuse modulatory systems in the brainstem are influenced by the amygdala. So this is just talking through some other things we don't need to do, but again, they're there for you. And I'm going to go ahead and call this good. So if you guys have any questions, I know we're just about out of time. So it was kind of a massive topic, my friends.
That was absolutely incredible. Thank you so much for taking the time to talk about this massive topic in such detail, as well as in an engaging way. I think these things are very obviously specific, and just, I'm always going to remember the swan. We're not doing hot dogs anymore.
No hot dogs.
Stuff. The hamster wheel. I really think it's a really nice learning tool. And obviously your passion for learning and teaching others neuroanatomy is really just evident. A couple questions. in addition to watching your lecture, this lecture, morning, afternoon, and evening, are there any other resources or anything else that you think would be helpful for students to learn neuroanatomy, or whether, do you suggest taking more of a structural approach, a network approach, or anything like that that you can provide?
I think, and at this point, honestly, I probably should try and I have a atlas that I've made that I actually is pretty simple. It's just not really available at this point. I do have a PDF of it that I would be willing to share. I think to me, neuroanatomy, it go in and learn the structures and then learn how they interact, learn how they're connected to each other. And then really in terms of kind of where psychiatry and psychology is at this point, it really is, we looked and yeah, there's these kind of neurological, we've got specific sites that cause specific problems. But in terms of these behavioral issues, they really are network imbalances. So issues, OCD is very much just a network imbalance. Depression is you basically have too much internal focus. Problems, as we age, we get different network activities, more so than specific regions that have a problem. So I think the first step though is the anatomy, and you just end up, I have to do so much like kind of flipping back and forth between things. And I really, really strongly suggest learning things relative to each other. So that's why I say like lateral ventral, start with the midline things, start with things like lateral, like holes. I always say brain holes because it's funny, but ventricles is more polite. But start with those structures that are easy to identify and then kind of work your way out. So if you'll work your way from the third ventricle out to the insula, and if you know relative orientation, then when you're kind of have something thrown at you that isn't familiar, you can usually find like kind of one lifeline to hold on to and then work your way to whatever you're trying to figure out.
That's really great advice. So start with the structures, really get that foundational, move to those neighborhood areas and start to connect everything then in terms of from a systems approach and how you understand disorders. And then review obviously the visuals like you've done a very nice job of outlining for us. So that'll be really helpful as well. And if you're able to share that and whenever you publish that, let us know. We love any resources for all the learners that are supported by KnowNeuropsychology. But thank you so much for so much of your time. And we're so happy to have you and to have learned from you today.
Yeah, it was definitely my pleasure. Thank you guys. And if anybody ever has any questions, I'm always delighted to get emails and answer questions. So just let me know.
Thank you so much.